started. I wanted to welcome you all to our inaugural PATRIC webinar. We really appreciate your joining us today. This is our very first webinar, so if you experienced problems or had issues with hearing or anything you think that we can improve, please let us know afterwards, because we are going to start doing these. We are thinking maybe once a month or twice a month, just to step through our PATRIC services and show people how to do different things within PATRIC. For the first one, we're going to be talking about a pretty new service that we have in PATRIC that involves building phylogenetic trees. The program or the pipeline that we use to do it is one that is very similar to other things that we have in PATRIC. We generally take well-established and well-known tools that bioinformaticians are using all the time and string them together into a single pipeline that allows researchers to come and easily do something that they generally, a bioinformatician or a computational biologist is generally required for a lot of the researchers. So the program itself was part of a dissertation here at Virginia Tech, and it's called Phylogenomic Estimation with Progressive Refinement, and it's called the PATRIC Pipeline. And I'll be describing that in detail today. Let me point out that when you run this pipeline in PATRIC, it's got set parameters and it has specific restrictions in PATRIC. However, if you're a bioinformatician or a skilled computational biologist and you want to use this to run larger trees, all of the trees in PATRIC, all of the order level trees, sometimes that contain hundreds of genomes, we run it using this pipeline. You can contact me if you're interested in getting the pipeline. It's freely available on GitHub. But what I'm going to show you today is how to run it in PATRIC. Well, before we really get into talking about the pipeline itself, I wanted to discuss a little bit about sequence-based phylogenies for bacterial genomes. Every workshop I give and a lot of email questions I get are from researchers that use PATRIC that have been interested in building trees, and they're interested in specific trees. Well, the questions I get most frequently are for ribosomal RNA trees. Also, I get a lot of questions. People want to build MLSA trees, multi-locus sequence analysis trees. Currently, what we have in PATRIC are protein trees that are based on the whole genome, the whole proteome across the genome. And also, our users are very interested in SNP trees. We now have the variation pipeline in PATRIC, and while we don't, at this exact moment, allow you to build a SNP-based tree, that is something that's planned for a future release in PATRIC, so it's coming soon. So let's talk a little bit about the ribosomal RNA trees and the phylogenies that are constructed on them. Ribosomal RNAs were first determined to be a really good gene to examine phylogeny. Carl Woese did this, Woese and Fox did this in 1977, and it's been determined that the 16S ribosomal RNA subunit is the most conserved, and it is a really good gene to use to build bacterial phylogenies. Now, this was before we were sequencing whole genomes. The 16S sequence now is just a single gene when you've got thousands of genes to examine in a phylogeny, but it still has its uses. 16S is standardly used when you have a culture or you don't know what the bacterium specifically is. One of the first things you look for is the 16S to try to get it down to genus or family. It's also had renewed interest with all of the metagenomic studies. A standard metagenomic analysis is surveying the 16S genes that are found in all of those reads. So 16S is very important right now for metagenomic analyses. But once again, let me point out it is one gene. And now that we are doing a lot more whole genome sequencing, there are better ways to look at relationships among organisms. Right before we got to the predominance of whole genome sequencing, a lot of research was geared at for determining relatedness among organisms. They would use MLSA, which was multi-locus sequencing. They would pick 7 marker genes, which are about 500 base pairs in length. You would amplify them up using PCR, and then you would combine the sequences together into a single long string and use the amino acids within that sequence to build the tree. Now, MLSA, we get a lot of questions for that. A lot of people are very interested in doing those kind of trees. We don't currently allow you to do that in PATRIC because we're doing the whole genome approach. But the MLSA is still widely used, and people are generally using it to identify organisms down to species or subspecies. But now that we have this tsunami, this cornucopia of bacterial genomic data out there, researchers are now going for a multiple protein approach, or really a multiple proteome approach, to try to take not just tens of genes, but thousands of genes and stitching them together and building phylogenies based on either the nucleotides or the amino acids behind those. So here's basically how the process works in PATRIC. You will choose a set of genomes. And from those genomes, we'll take all the genomes together and we will build protein families. And we will look specifically at protein families that each genome that you have chosen has at least one protein as a representative of that. When all those protein families are brought together, we concatenate them into a single string, and we build the tree on that. Now, we also include support values for those trees, which I will describe later. So basically, from your perspective, on the left side, the circles denote the genomes. Those are the genomes in. That middle part is all happening behind the scenes in PATRIC, but what you'll get is the tree out at the other end. So PATRIC's tree building pipeline, or the PEPPER pipeline, has a number of components within it. And I've listed all the components here and the citations for those. Any of you who have attended a workshop or have looked at the PATRIC site, we have a list of tutorials, which I'll show you how to get to later. But at the end of the tutorials, I list all of these different parts of the pipeline with the citations, and I do this for a specific purpose. If you're building these trees and you're publishing, you need to cite your work, and this is how we hope that you will cite it. You'll say something like, I built the phylogenetic tree in PATRIC, and please cite our primary citation because we live or die based on how many citations we get. And then within it, after that, it can say, this pipeline involves these specific components. So it's all there for you. We want to make it as easy as possible for you to publish using the PATRIC resource. So PATRIC, the PEPPER pipeline in PATRIC looks pretty complicated. This is what's happening behind the scenes. And I'm not going to go into all of this because it's actually looks pretty boring except for the people who are interested in how pipelines work and stuff. So I don't want to go into all of the components of it, but I want you to pay special attention. I want to go into to define this specific part of the pipeline, way over there on the right side in the middle of the page. After you get a fast tree, you can refine it. There's a box there that says refine. Now, the tree building algorithm in PATRIC has something special that called progressive refinement that I'm going to talk about now. So what progressive refinement does is when you're first building a tree and you select all your genomes, so look across the first row of this diagram, and you see there's a large number of genomes on the left side that are denoted by circles. They run through the pipeline and it spits out a tree. If you have selected progressive refinement, what that does is it looks for branches of the tree that are weakly supported. When it finds one, it will take all the genes within that branch. On the upper right side, there's the little box around part of the tree that's supposed to denote that part that is weakly supported. It's going to take that subset, and what it's going to do is collect the genomes only in that subset and relaunch the pipeline for that. Now let me point out that when in the first row, when you're looking at all the genomes, one of the requirements of the pipeline is that it will only use protein families that every single genome that you've included has a member within each family. So those are the only families that are included. You can imagine that there might be a lot of families that are specific to a particular clade. Perhaps there was a large lateral transfer that has been passed down to all the members of a specific clade of the tree. That won't be included in the first round of the tree. However, if that branch is poorly supported and that particular clade is selected for progressive refinement, you could conceivably bring in more protein families to add more support to those specific genomes within that tree. So it'll create this, let's call it a subtree. And then this subtree, the pipeline will insert the subtree back into the main tree, replacing the weak branch with the next branch it is made. Now there's a caveat here. You would think that it's going to be 100% support, but that is not necessarily the case. Although in most of the times the support values will go up, sometimes after progressive refinement they could even go down depending upon what the components are for that particular part of the pipeline. So we've covered the middle row. After it's identified the first branch with weak support, it goes down to the next branch to see what the support values are in that branch. So Pepper, the pipeline, the tree building pipeline, will step through each branch to try to increase the support values at each of the branches. And each time it builds a new subtree, that tree is inserted into the final tree. So basically, what it's doing when you engage progressive refinement is you're identifying trees that have less than 100% support values. So it's identifying the subtrees, or you might want to call it the branches or the clades. One particular aspect of this pipeline is that specific subtree has to have the next branch of it, the next lower branch, have 100% support. When it finds that, those characteristics, it will take all those genomes and relaunch the pipeline. And it'll relaunch the pipeline choosing the next part of the tree, the next branch down as the outgroups. Once it has generated the subtree, it will insert it back into the tree, replacing the previous branch that was weakly supported with the new branch. The tree pipeline will continue in this process until each of the poorly resolved branches that meet the requirements have been examined. Now, it's very cool and it's a very powerful aspect to the tree, but let me warn you, if you choose to use it, it can be very time-consuming and expensive. what might take hours or a single day to build a tree can extend on quite a long time. So what I would suggest is the first time you run the tree, run it without this particular option selected, because you may have a tree with perfectly supported branches and you won't need to do this, and then you won't need to wait for it. So we're just going to show you, or I'm just going to show you what it looks like. So here's a tree. This is actually from the Rhizobiales, an order of bacteria. Yes. So here's a poorly supported branch. And you can see that the next branch down, it's strongly supported. When you don't see a number in any of the PATRIC trees at a branching point, that means it has 100% value. So what Pepper will do is take all of the genomes within those two branches, the poorly supported and the strongly supported branch, and build the tree with those genomes using the next branch down as the outgroup pool. So that's probably more than anybody wants to know, right? We just want to show me where to put my genomes in and give me the tree. That's generally actually how I want it to work, but sometimes, you know, there are some of us who want to know the nitty-gritty details, so that's how it's working. Oops, I went the wrong way. And this cartoon is showing you at each of the steps where the different tools are engaged in building the trees. So I wanted to talk a little bit about the support values and how we, what support values we use in building the trees in PATRIC. This is something I get asked all the time. People automatically think that we're doing bootstraps, but actually we're not doing bootstraps. I'll describe the three major ways that people are using support values for trees. So you hear bootstrapping a lot. That's a procedure where you take a random subset of the data and then you rerun the phylogenetic analysis. The reported value and the percent of the bootstrap indicate, it replicates, it'll show you in how many trees had the same value at that node. So in 100 in bootstrap terms means that the same node showed up in all of the bootstrap replicates. So that's kind of an expensive way to add value to a tree. Jackknifing is a different kind of support value. It's similar to bootstrapping, but rather than resample all of the data, what it's doing is taking a subset of the data and what it's trying and rerunning the trees. You can imagine that. Yes. Question? I guess not. Okay. You can imagine a situation where there could be a group of protein families that could have undue influence on a tree. They could be way off, and jackknifing is a way to look to see if specific protein families are messing around with the tree, to put it, if there's an outlier character that might have a disproportionate influence on the relationship. So jackknifing is a way to get around this, and jackknifing is actually what we do. There's another method you can use, which is gene concordance, which is looking at the individual gene trees and seeing how strongly they agree with the overall species tree. So PATRIC is using the jackknifing, and those are what the support values are on the tree. It's taking 50% of all the genes and rebuilding the trees, and it creates 100 topologies for each of those, and then it's looking to see how many times the same branching pattern was seen when they ran that with 50% of the genes and the topologies. It's quicker than bootstrapping, and that's one of the main reasons we like it, but also jackknifing, a lot of people like it anyhow. Okay. So, any questions about that before we get on to today's example? You can either text it, we have someone following the text, or you can ask out loud. Okay. So, today we're going to do an exercise that involves building trees with Mycoplasma genomes. Mycoplasmas, a genus that lacks a cell wall and a membrane, and because they don't have walls, they are not susceptible to many of the antibiotics like the penicillins or the beta-lactamases that target cell wall synthesis genes. They also have small genomes, and the tree should build pretty quickly. I say that the tree should build pretty quickly, but one of the things in PATRIC is it's all based on a queue. So these trees can build very rapidly, but you may be waiting for a long time in the queue to build your tree. I just submitted some trees recently, and while the trees, I tried 2 of the different tree building algorithms, and with progressive refinement and without progressive refinement. So I submitted 4 identical trees with each of the different parameters I could choose. They ran from between an hour to 4 hours, so that was really fast, but I had to wait for 3 days because the queue has become quite popular. So let me caution you that this service is gaining in popularity and right now we're trying to figure out how much computational oomph we have to put behind it. So we keep increasing it, but the more we increase it, the more people are using it. So you may have to wait a while. You'll notice I'll show you how to do some certain things today, and you'll see that your job may wait around in queue for a long time. But that doesn't mean anything's wrong. It just means that it's going to take a while for the people to move ahead of you and clear out the queue so you can use it. So here's what we're going to do today. We're going to create a genome group that we want to build the tree on. We're going to submit this tree building job at PATRIC, and part of that is we're going to just identify an outgroup genome, and I'll show you how to find that genome specifically in PATRIC. I'll show you how to monitor the progress of your tree job, locate it once it's finished, and then we'll look at examining the results. I'll also show you some different tools. PATRIC will provide you with an SVG picture of your tree, but you may want to mess around with that a bit. So I'll show you 2 different pieces of software too that you can use to examine your tree. So what we're going to do today is we're going to use Mycoplasma, and we're going to have 3 genomes as our in-group genomes. You notice that, well, the 3 genomes are from Mycoplasma Genitalium. They're all pretty small. They're all a little less than 600 kb in total length, and they all have just under 600 CDSs, which are genes. The outgroup genome that we're going to use is Mycoplasma mycoides. And you may have some questions while you're working in PATRIC. How can I choose an outgroup genome? Maybe people don't know about the concept of rooting the tree and looking at specific outgroup genomes. So we're going to step over, we have tools in PATRIC, we have trees in PATRIC that you can use to look at what could potentially be, you could use as outgroup genomes, so we'll talk about that. There's one other thing I just wanted to mention because this comes up every single time I give a workshop. You notice that I have the names for the genomes, and then right underneath it, it says Genome ID with a specific number. Believe it or not, the biologists that are out there listening, Genome ID is a very important number. Let me give you an example. Right now, we have 15 Mycobacterium tuberculosis H37RV genomes in PATRIC. So that name is not specific. Why do we have that that many genomes with that name. Every lab that had that original culture stock, it's been in their lab for a really long time, and generally they're working on it and then they sequence it to see if it's changed, because of course they do change. There are a number of SNPs that occur every generation, and so you want to have the most accurate sequence for what you're working on, and you sequence it, you publish it with your paper, you submit it to GenBank, and then we get it. And so there are all these H37RV genomes. So the name's not specific, but that number is specific to that particular genome. So I just wanted to bring that up so everybody's aware of that, that sometimes you can use the Genome ID number to find a specific genome when there are multiple genomes that have the same name. So let's build some trees. Any questions while we set up here? Okay. If there are no questions, everybody should be logged in. If you're not logged in, PATRIC won't allow you to build trees. It's not that we're going to spam you or want anything from you. The reason we request that you become a registered user of PATRIC is that to use our services, that generally means that you have data that you want to keep private. So we have to create a private workspace for you, and there are specific restrictions, and we want to be able to ensure that your data is private and that you only have access to it. So that's why we request that you log in. So let's go into services, which is across the top, and you click on the tab, and there is the phylogenetic tree service. Now first we're going to create the genome group, but I want you to look at the page, and this is something new that we have in PATRIC. You see it's supposed to be books next to each other. That's an icon that if you click on that, it will take you directly to the tutorial in PATRIC that shows you exactly how to run the service or build the phylogenetic tree. So I just wanted to bring that out and point that out to you. Okay, so we were going to build the tree using 3 Mycoplasma genomes. How in the world can we find one of those? Well, up here, I'm going to increase this so you guys can see it. Well, maybe not that much. That's a little too much. Okay. In the global search box, Mycoplasma, I'm going to enter the term Mycoplasma. Now, and let's just see, because maybe you can't spell too well, maybe you don't. I was worried that I wouldn't be able to spell the name of the genomes that we're using today. But so I've entered that in the search box, which you should as well. And you can see that it shows me the search results. For some reason, it gives me antibiotics, specialty genes, genomes, taxonomy, genomic features, and transcriptomic experiments. Well, let's see, what's a good way? We could go into genomes, and genomic features is all the genes and stuff. But let's go to the Mycoplasma taxonomy page. We could easily go to genomes, but I wanted to give you some further experience with PATRIC. So let's all click on Mycoplasma together. Is everybody with me? If you're not, now's the time to shout out. So we don't want to leave anybody behind. Is that the Marines that say that? I can't specifically remember, but here we should all be in the same place. So we wanted to find, remember we were looking for 3 Mycoplasma Genitalium genomes. So I could go through taxonomy to find that and click on that and look for, scrolling slowly down to Genitalium. I could find it that way. to find those specific genomes. I could also go through the Genomes tab and write in Genitalium in the keyword search and search for those specifically. And thank goodness I spelled it correctly because I was able to find it. I'm not the best typist and not the best speller either. So we were going to build this with 3 of these genomes. Which 3? Now, I don't know if you guys can see the Genome ID. Mine has the Genome ID, but let me show you how to find that. So you see this plus button here over completion date, the plus icon? Click on that. And that will show you all the columns that you can add to this view. And you can see there are a lot of them. We collect metadata and try to provide as much data as possible for each of these genomes. I have clicked in the Genome ID, and you should click it in too, because we want to look at these specific genome IDs, or the names as well. And to get rid of this, I have to click the plus button again. Okay, I'm going to need to pull up this slide, and I'll pull it over to the side so that you guys can see it too. We want Mycoplasma, Genitalium, M2288. Okay, and that, so I clicked on that. And then we wanted M2321, M2321. Clicked on that. And then we wanted G37, G37. And I'm going to wait just a moment here to be sure everybody's caught up. If you have any questions, now's a good time to ask them while I'm waiting for everybody to click on those genomes. You notice that as I started clicking, this vertical green bar started, it had been empty before. Let me show you. Before there was nothing. And when I click on these genomes, you notice icons pop up. Those are indications of once you make selections in this table, it's telling you all the downstream types of things you can do with the data that you have selected together. You can download the data. You can copy it. You can group these things together and go to a genome view that looks specifically at the genomes that you have selected, or you can create a group. So let's do this last one. Let's click on the group icon in the vertical green bar. Okay? And when I click on that, it opens up a pop-up window that gives me a couple of choices. This first one says new or existing, and this text box says existing group, but at the end of it you see a down arrow. Let's click on this. So I'm assuming that none of you have made this group before, or you don't want to add data to an existing group, so we're going to click new group. I'm going to call it Mycoplasma Genitalium, and I'll call this November 30, so I know which group it is. So you need to click New Group, and then you need to give your group a name under the group name. And the reason I'm asking you to do this is PATRIC, you can add these genomes one by one in the tree building service, but grouping them together allows you at a later time to go in and look at that data more closely, but it also allows you to think about things as you're doing it. And the tree building pipeline allows you either to look at individual genomes or to look at genome groups. So once you've done both of those things, let's click the Add button. And once it's clicked, down here in the lower left column, it will give you a little ephemeral call it an ephemeral pop-up box that tells you that it has successfully created a group for you. So a little earlier, we were talking about in-groups and out-groups. We've selected this to be our in-group genomes. Now we need to select out-group genomes. In the tree building pipeline, you can select out-group genomes that can have between 1 and 5. You have to have at least 1. You can't have any more than 5. But you may be wondering, how do I choose an outgroup genome? I don't even know what that is. So over here, we have the phylogeny tab under this Mycoplasma. So let's click on that, everyone. And this will show you these are the same, these trees. I'm going to shrink this down a bit so we can see them more easily. And it may be small for you, but if you have it on your own computer, you can see it as well. It shows you these are order level trees that we build in PATRIC, and you can see there's a fair number of genomes here as I scroll down, probably maybe 200 genomes. So I can see Genitalium here, and if I couldn't find it, I could do a search for it, Genitalium, and you can see it's been highlighted on the tree. So when you're choosing an outgroup, you want to choose something that's below the group that you're choosing. So look, all the Genitalium here are all sorted together in a nice, neat group. I could choose Mycoplasma gallisepticum as an outgroup, or Uriplasma urolithicum. And look at this. So we have Mycoplasma here, Uriplasma here, and then back down to Mycoplasma. That indicates to me that there's some incongruence within the taxonomy that probably needs to be resolved. Either Uriplasma is, in all actuality, perhaps a Mycoplasma, or everything below this point is not Mycoplasma and perhaps it's something else. When you see that kind, to me that's always very interesting and things I want to more deeply explore. They were probably named for some phenotypic character early on, but it's been maintained just because it's got a research community that really loves the name Ureaplasma. So in the original example, it was Mycoides. Control find Mycoides. So there are the Mycoides down at the bottom. So the person who originally was building this tree chose Mycoides as the outgroup. Conceivably, any of these up to probably the Uriplasma. I personally probably would have chosen the Uriplasma, but why not? The Mycoides is a good solution for that. But any of these below probably this branching point could serve as a credible outgroup for any genome that you want to have, as any outgroup you want to choose. Now, this was Mycoplasma, Mycoides, and it was JCVI for J. Craig Venter Institute. I know they did it now, and that's this SIN 3.0 was the genome number. Okay. So let's go back to the tree building. Let's go back to the tree building pipeline. I'm going to make it a little bit bigger now so you all can see it. And I'm choosing phylogenetic tree. Now we're actually going to build the tree. I know you've all been on the edge of your seats waiting for this auspicious moment, and here we are. When you look at this, there are 1, 2, 3 boxes. Each box has something that you have to do before we can submit the tree, and when all the data that the pipeline needs to run is assembled, this button will turn blue. So it's a good indication of something you failed to do if it doesn't. So remember, we had created a group to build these trees with. So to find that group, in groups under and/or select genome group, there's the text box with the down arrow. Click on the down arrow, and it's going to show me the most recent group I created first. Now, let's say you want something different, like if it's an old group, I do a lot of Brucella work. It might not be, look at the names I give these things, pretty embarrassing. But you can also do that, just start typing something to try to find a group that you're interested in. But right now we know this group, we just created this, we're desperate to know about this particular, how these trees are all related. So let's click on that. Then once we click on it, we're not done yet. You see this plus icon here? We have to move this group into the in-group genome table box to be included. And the reason we have this is that you might have several different groups. Maybe you want to build a series of trees, one for individual groups and then combine them all together. So you'll need to move them into this one at a time. So if I click on the plus button, now that group is in the in-group genome table, and you can see it's got the 3 genomes here. So if I were to think I could submit the tree, I can't. It's still gray. I've got some more work to do. To find the outgroup, it was this Mycoplasma mycoides JCVI SYM 3.0 genome. Oh my gosh, that's such a long name, and I'm sure to mess up all the spelling of it. But in this text box under Select Genomes, I'm going to start typing that last thing that seemed to be pretty unique. See, I didn't have to go too far. I got that SYN 3 point, and below it you can see in the text box the name of the genome I was searching for. Also note that it's got the genome identifier behind it, so I could have also used that to find that genome. So I click on that, and then what do I need to do? I've clicked on it. Can I submit the genome? No, not yet. I have to add that to the Select Outgroup genome table. So let me point out we've done 2 different things here. In the in group, we added a genome group. In the out group, we added a single genome. You could also do that for the in-group genomes if you wanted to. If I wanted to add a Genitalium, now my bad spelling might come back to haunt me. Oh, look, so I've got all these choices here, and maybe I could choose one. Let's say I wanted to add this one to it, and I added it as a single one and added it in. And then, oh no, I have buyer's remorse. That's not what I wanted at all. Do I have to relaunch the whole thing to get rid of it? No, I can get rid of it simply by clicking on this X. And then I'm back to that beautiful group of 3 genomes for my in-group, 1 for my out-group. Now you may be wondering, how many genomes can I add? Right now, PATRIC is set up to 50 genomes in the in-group and up to 5 genomes in the out-group. If you ask us nicely, sometimes we'll work things out in a different way for you. So I've got my in-group genomes and my out-group genomes. Can I submit the job? Not yet. I have to put the job in a folder. So now we have to go down to parameters and do things. I've already created a folder that I want to house this in. But if you haven't created a folder yet, you can click on this folder icon, which will open up a pop-up window that will show you all the folders that you have in your workspace. If you want to create a new folder, you go up here to the upper right, click on Create Folder, and I could call it Mycoplasma folder. Actually, I don't want to create that folder, but if you wanted to, you could create, I'm going to call it a webinar phylogeny. I could create the folder if I wanted to. And then once it was created, I'd have to scroll down and find it, webinar phylogeny, click OK. and then it appears there. You see, and once you've done it once, you'll also see it in the down drop box. It also needs a name: Mycoplasma, Genitalium, Mycoides. That's what I'm going to call this tree. All right? At this point, I could submit, but first let's look at this. There are two tree building pipelines that you can use: FastTree and RaxML. Both are well-documented, well-liked pipelines. FastTree is like its name, fast. That's why we have it at the default. So I'm going to keep it at that. And you can do progressive refinement, yes or no. For this first tree, we're going to do no. And then we're going to submit the job. Okay? And once I submit the job, there is a pop-up message that tells me that the job has been launched. Okay, so this tree is going to build. We're not going to see it today, but I can show you some other trees, that I have built so that you can see what happens once you build a tree. So the first thing is, how do I monitor the progress of my tree? Down here in the lower right, you see this Jobs button. Click on that. And for those of you who are new to PATRIC, this is new and it's pretty cool. Well, the jobs page isn't new, but up here I can filter on all the different jobs I've run, and I only want to see my phylogenetic tree jobs. Here is the job I'm currently running. It's queued, and here's another one that's queued, but these are the jobs that are completed. So that's the way you find out the status of your job. Also, if you go down here and mouse over it, it'll tell you if it's queued, if it's running, or if it's completed. So let's say I want to see one of these jobs I ran recently. I have this Bartonella job that I liked a lot. How do I look at that job? Well, you can click on it, and notice that once I click on it, it highlights some things in the green bar that allows me to look at it more carefully. I can view the job, or I can report an issue. If I had a failure on a job, I would click on this. It would open up a thing that would immediately send an email saying that this job failed, and you can add some text in here if you think there's, something wrong with it. Like sometimes we get things like, this job has been running for, it's still been queued after 5 days, what's going on? That's okay, you can submit that kind of stuff. However, if I want to view it, I click the View button here, and it takes me to this page with a whole lot of stuff that you may not necessarily want to see. But one thing you can see is that this was a different job I ran that had 20 genomes and 5 outgroup genomes. And that took an hour and 17 minutes to run. You'll also notice there's a whole bunch of stuff down here. The only thing that I think that you would need to have at this point is the NUIC files. The final NUIC is the tree with the genome IDs, and this regular NUIC has the genome names. So if you want to take this data and load it into a different program to view the trees, you're going to need the NUIC file to do it, and you could just click on it and download it. And my computer asks me where to download it, which is I've got it under this webinar folder, so I just put it there. But I really want to see the tree. I can see the tree. Let's click up here where it says View Tree. And this is the tree I built for Bartonella. And you can see that there are a lot of branches here with no values. That means those branches had 100% support as it was, so they wouldn't even be considered for progressive refinement. But there are a few branches here with less support. So actually it would take this branch with the 38, because the next branch out has 100% support. And so all of these from Claridge all the way up to Hensley would be included for progressive refinement if we were to choose to had chosen that option. And the APIS strain would be the outgroup. you can go in and you can, let's see, there should have been, I have to go back. I can see it as a cladogram too over here in the corner, but I personally like the phylograms, so I'll keep it at that. Now, I showed you how to download the Newick file. There are two programs that provide a really cool way to look at phylogenetic trees. I mean, this is fine enough, and you can download an SVG of this as well. But Figtree and the Interactive Tree of Life are both places where you can upload your Newick file, and then you can adjust it. And I have one here of this same Bartonella group. This is what I uploaded in Figtree. And what I like about it is I can turn things on. So I chose to put the support values here, and I can change the font size on them that I want them to be. And I can look at the tip labels, and I can make the tree bigger. I just really like this. And also there are certain ways I like to see trees. So I can click this, and I want to rotate this branch because I think it'll look better this way. And I want to rotate this. So a lot of the things, you know, people like to see trees in specific ways. With Figtree, you can go in and actively manipulate a tree and then download it as a SVG, a PNG, or a JPEG. The Interactive Tree of Life, if you have metadata associated with those genomes, you can upload the metadata with the tree, and it'll give you a tree that shows you the metadata along the side. So I've been talking a long time. We've talked about building the tree. I've shown you how to create the groups, how to launch the jobs, how to monitor the progress, and the files that you will need to download if you want to use it in a different program, and how to view the tree. Are there any questions that you have that I can help you with at this time?